children, we have studied RH blood group system in our previous video lecture, right? We know that person who have RH antigen on their RBC surface, they are considered to be RH positive. Person in which RH antigens are absent on their RBC, they are known as RH negative. We also know that RH negative person cannot receive blood from RH positive person. If blood from RH positive donor is transfused to RH negative recipient, that RH negative recipient shows immune response against the RH positive donor RBC. This is because immune system of RH negative person, thus RH antigens act as foreign antigen or alien antigen or non-self antigen. As a result, RH negative person produces anti-RH antibodies, right? Thus, anti-RH antibodies secreted by RH specific plasma cells and also RH specific memory B cells produced. This RH antibodies will recognize RH antigen. See, this is RH antibodies easily recognize the RH antigen. Then it binds to the donor RBC. Right? This would result destruction of donor's RBC. If there is any subsequent RH positive blood transfusion means, right, if the person RH negative person receives the same RH positive blood again, what will happen? It will also get fail easily in recipient body. This is because the recipient has been exposed RH antigen in their previous transfusion itself. Therefore, anti-RH antibodies and also RH specific memory B cells are present in recipient body, right? When the RH antigen encountered again for the second time, what will happen? The memory B cells get activated and rapidly produce anti-RH antibodies. Besides the blood transfusion, RH incompatibility is seen in pregnant women. Besides the blood transfusion, RH incompatibility is also responsible for complication in pregnancy. RH incompatibility in pregnant woman results hemolytic disease of the newborn, short form HDN, right? RH incompatibility is also responsible for the complication in some pregnancy. It results a disease which is named as hemolytic disease of the newborn, HDN. What is HDN? HDN means blood disorder. It occurs in the fetus. When it occurs? It occurs when the blood group of mother and fetus are incompatible. Right? It results following some symptoms. They are fetal RBC hemolysis, hemolytic jaundice and anemia. Right? We'll start discuss in detail about HDN or erythroblastosis fetalis. As we already know, most important blood group system after ABO with respect to blood transfusion, right? Let us see two parents who are heterozygous. The Rh factor, that is, they have one positive and one negative allele for the Rh factor. If we work out this with the checkerboard method to find out the possible genotype of parents and their offsprings, we will get this kind of result. The results are, the genotype is plus plus, plus minus, plus minus, minus and minus. From this we get three different genotypes, that is positive, then positive negative, then minus minus genotype, right? Homozygous positive, homozygous negative, then heterozygous positive. These are the three different genotypes we receive as a result. However, we only get two phenotypes, that is Rh positive, Rh negative. We may get three types of genotype. Genotype means genetical makeup, that is homozygous positive, homozygous negative, heterozygous positive. That means one positive, one negative, right? But the phenotype will be in the form of two. One is Rh positive, which is coded by D, capital D, and Rh negative. As the result, Rh positive acts as dominant allele. Always Rh positive allele acts as dominant allele, and then Rh negative is a recessive allele. Rh factor is inherited like Mendelian fashion, because dominant allele, that is Rh positive, is masking the negative recessive allele. In a blood sample, if you add commercially prepared, this is actually a commercially prepared blood grouping kit, what it results, antibody against the Rh factor, that is antigen D, and you end up getting agglutination result, right? This result shows Rh antigen is present, that is D antigen is present in the blood sample. Got it? That person is Rh positive person. However, if there is no agglutination in the blood sample, this means that Rh antigen or D antigen is absent. Thus, the person is Rh negative. Shall we recap this blood sample test again? In a blood sample, if you had commercially prepared antibody against the Rh factor, what will happen? That is antigen D. If we add the commercial antibody in it, if it end up, the agglutination means what it results? Rh antigen, that is D antigen is present in the blood sample. So that person is Rh positive. However, if there is no agglutination, the blood sample shows without clumping. So, the Rh antigen or D antigen is absent. So, the person is considered to be Rh negative. As we discussed earlier, beside the blood transfusion, RH incompatibility play a crucial role in the pregnancy of the RH negative mother. Consider that a RH negative female, see the picture, RH negative female marry a RH positive male. In negative phenotype, only one possible genotype will be there, that is minus and minus, mother genotype, right? However, for a RH positive phenotype, this is RH positive phenotype, in dad's body, there are two different possibilities will be there. The father can either be a homozygous or heterozygous. The father may be a homozygous father or heterozygous father. Homozygous father means the RH factor, that is plus genotype is present. If the father is heterozygous means the genotype will be plus and minus. If the father is heterozygous, see the first checkerboard. If the father is heterozygote, heterozygote means RH positive, RH negative. So that is heterozygous condition. If the father is heterozygous for RH factor, there are 50% of chances of having RH positive offsprings. So they may produce 50% of RH positive offsprings. As we work out with the help of the checkerboard, that RH negative mother with heterozygous father, RH positive and RH negative, what it results? Plus minus, minus, right? So you get two genotype with plus minus, which will give you RH positive, right? What about the rest of the two genotype with minus will give you a RH negative phenotype. Thus, out of 4, 1, 2, 3, 4, out of 4, there is a 50% of chance of getting RH positive child. That is in the heterozygous condition. Got it? However, if the father is homozygous, see the checkerboards 2. If the father is homozygous means positive. When the father is homozygous, what will happen? It results 100% chance that all offspring will be RH positive. As you observe from the checkered board, since the father is plus, see, the father is plus. Every child will get one allele from the father, one allele from the mother, right? So that's why all offspring are RH positive. If we don't know whether the father is homozygous or heterozygous, right? We don't know the exact result of the father's blood group. The father may be a homozygous or heterozygous. We can consider that there is 75% of chance RH positive offspring will be there. Will turn out to be a RH positive child. Observe the checkerboard carefully. We have 4 RH positive here plus 2 RH positive here. So how many? 6 out of 8. That is around 75% of offspring will have the phenotype of RH positive. Got it? In 25% of cases, 25% of the cases, RH negative mother will conceive RH negative child. In this condition, there are no issues at all, right? But let us consider an example. When RH negative mother, see, this is a RH negative mother will conceive RH positive child which contain RBC with RH antigen, right? This RH antigen are present in the child. However, since the mother is RH negative, they don't have RH antigen. During delivery, the RBCs with RH antigen from the child can enter into the mother's system during the paturation time, childbirth time. But in mother's bodies, RH antigen will be a non-self or foreign antigen because mother is RH negative. Mother is actually a RH negative. So they considered RH positive as a non-self antigen because mother is RH negative and they don't have RH antigen in their body at all. So the RH antigen of the child is treated as foreign antigen or non-self antigens by the mother's system. As we know, the cells of our immune system can differentiate self and non-self antigen, right? When our immune cells come to contact with the RBC that expressing self antigen, they will never show the immune response, right? As our immune cells are moves around the body, it may meet up with the RBC with expressing self antigen. At the time, it will never show the defense mechanism, right? Because they carry self antigen. However, immune cell may meet the RBC with the foreign antigen, right? What will happen? Surely it shows their defense mechanism against the foreign or non-self antigen. Thus, immune cells of mother's body produces antibodies against the antigen D. If the baby blood is RH positive, right? Then the mother body produces anti-D antibodies. Thus, immune cells of mother's body produces antibodies against the antigen D or RH antigen of the RH factor. The mother will prepare specific memory B cells. See, the mother body will prepare the specific memory B cell. Recall the blood transfusion. The mother will prepare the specific memory B cell. Will preserve memory for this particular antigen for her lifetime, whole lifetime. Thus, memory B cells produced by mother's immune system. This memory cell will recognize the particular RH antigen throughout her lifetime. In case RH positive antigen comes in mother system, imagine this is a mother system, right? When the RH antigen come in contact with the mother's system, what will happen during the time mother system shows immune response? In case RH positive antigen comes in mother system, for the next time the mother is prepared with the antibodies to kill the RH factor easily and fastly. During the second pregnancy, what will happen? RH positive child, as we already know that 75% of chance of getting RH positive child again, right? At the time, the memory B cells which are already produced from the mother's system convert the plasma cell. They convert the plasma cell into antibodies. Large number of antibodies. Right? That antibodies start to fight against the Rh factor in the baby's RBC. We know that during the pregnancy, mother and child are connected through the placenta. So this is the placenta, right? Why they are connected? Because child depends on mother for its nutrition. However, placenta only selectively allows the substances. Placenta only picks, this is the placenta, only select the some substances which can pass through the placental barrier. Out of all type of antibodies, IgG antibodies have the capacity to cross the placental barrier. Right? Out of all the antibodies, IgG type of antibodies can cross the placental barrier from the mother system. This type of antibodies can enter into the system of the child. IgG produced by the mother system. Right? IgG antibodies which is responsible for the Rh antibodies can cross the placental barrier. Then it enter into the child system. When entering into the child system, these antibodies will attack Rh positive blood cells of the child. Results child death. Thus in this condition, for the second pregnancy, the mother can only give the birth to a live child if a child is Rh negative. As we already studied, 25% of chances of getting Rh negative child. If there is Rh negative child, there is no problem at all. If the child is Rh positive, it may 75% chances there. Definitely the child will die. This can be prevented with. How can we prevent this? This can be prevented with ROGAM. ROGAM is actually a anti-Rh antibody. Before the mother immune cell produces antibodies, what will happen? This ROGAM injection administered by the medical practitioner to the mother who is having Rh negative blood, right? It will stop the activity of the memory cells which is ready to make the antibodies. It finds out all the Rh positive RBC and destroy the RBCs of child immediately after the first delivery. After the first delivery, what will happen? When the babies come out of the mother's body, at the time baby's RBC mixes with the mother blood, right? So doctors immediately administered the ROGAM. That ROGAM have the capacity to destroy the RBC which are produced by the child. Got it? Yes? See? How to prevent? Anti-D antibodies administered to the mother at 28th or 34th week of gestation. If mother negative delivers the Rh positive child, the administration of anti-D antibodies done soon after the delivery. So it develops passive immunity. It prevents the formation of anti-D antibodies. It prevents the sensitization of mother's immune system. So this ROGAM avoids the mother's sensitivity. In the mother, there is no Rh positive RBCs. Thus, the mother will not create any memory cell or antibodies. Since the mother is not having memory cell during the next pregnancy, the mother cannot create IgG antibodies, which will go into the child system and destroys the child RBCs. Even if the mother is pregnant with Rh positive child, she can successfully deliver the live child if the doctor administered the ROGAM injection. I hope you can understand the concept very well. Thank you, children.